So that's recording. It's recording on a cloud.
Think about it. Why you got a problem? Do you like it? All right. At worst, I mean, there's two reasons. One is the statistical approach you use to grant that, which I don't like.
If it's recording to the computer, it's.
And that's why I just want a general point. Can we talk after? Oh, I don't know.
That's all right. There we go.
So it is recording.
All right.
Cool. Yeah. And then we can edit it from the link.
Let's
sounds good.
Maybe we can chat on the phone later.
Thanks.
All right. Thank you, Liz. All right. Wicked. Okay. So we have some new faces and then some missing faces. No, not enough engineers, Joe. Okay, good. Should I stall? Okay, good stuff. Okay. All right. So round two. Let's see if this works. Yeah. All right. So I've been told I'm not allowed to use the whiteboard now because they're now recording my voice in the slides. So please ask whiteboard questions so I can punish all the people that couldn't be here. All right. So today, what I thought we'd do is a sort of quick whirlwind recap of the kind of highlights from last week or 2 weeks ago. The recordings are online or have been emailed. Right? Did people get a link for that sort of come out? Yes, no. If not, we'll check at the end of it. We can kind of put it all together. And what you remember as well last time, one of the key take homes is that hearing people talk about neuroanatomy is really not enough. It's not going to do it for you. Part of it is getting a worked example, being like, oh, I've seen this paper. What on earth is that region? How does that fit within the context of these things? And really going after the problems yourself with the sort of a question in mind, because otherwise it all just seems like a bunch of facts that as soon as you sort of like turn a corner, they all just drop down and all the structure goes away. So if you really want to kind of get this stuff, you need to kind of dive into it on your own. All right, so recap with a clicker that's not working. So let's do this. All right, so the recap. So the starting point, we talked about the fact that brains are kind of oddly shaped. And so up and down and left and right and forward and back don't really mean an awful lot. So you need a reference point. So we have things like superior and inferior. We have caudal, which is from the tail end to rostral, rostrum, the nose end. And we also have dorsal for the dorsal fin or ventral for where the vents are. And we can talk about things in that space and you'll often hear those spaces. So if you get confused, just Google, there's a billion different images. I'm going to start from the opposite point I started from last time. So we've got this organizing principle in the spinal cord, which is that the posterior regions are typically and the lateral regions are carrying sensory information input up the spinal cord into the brain, and the motor regions are happening in the sort of lateral and more anterior sections, are shooting it down, and that's all in the white matter because there's myelin wrapped around the axonal sheaths to make them a little bit more efficient and quick. In the middle is the gray matter, these horns of the spinal cord, which is where all the interconnections and interneurons of the spinal cord live, which are already doing some integration of the sensory and motor signals to put them into little paths together so that you do things like coordinate muscle extension and flexion at the same time. All the way along, we have these different sections that are split up into body segments. So cervical for neck, thoracic for body, lumbar for the hips, sacral down to the bottom of the hip bones, and the coccygeal is our little, what used to be our tail. And you can see all these nerve roots falling out that are pulling information in and pushing information out from the brain as it controls and manipulates these different affordances that exist within the body's interaction in the world. This is a little cartoon diagram of the brainstem. So the spinal cord here would be running all the way down. The most superior bit is the midbrain, which contains a few cranial nerves and a lot of other really interesting neuromodulatory structures. We talked about the pons, which has one of my favorite areas, the locus coeruleus sitting in it, but also a lot of little nuclei here, the pontine nuclei, which are receiving inputs from the cortex and then projecting into the granular layer of the cerebellum. That will come up again today. The medulla has a lot of more homeostatic regions. The vagus nerve, which is sensing all the activity in your gut, comes in here. And synapses with the solitarius nucleus, which is really important for communicating visceral functions to the central nervous system. On the right-hand side was just a little diagram of the autonomic nervous system. Easy to forget, but very, very important for defining the homeostatic principles in the body. I'll talk about this one a lot more next week, but I just sort of kind of want to keep this in our minds. There's these really interesting structures sort of smeared through the brainstem and up into the basal forebrain that are controlling the volume knobs, the gain on the system. We call this the reticular activating system because when you looked at it under a microscope back in the old days, it looked like a net that was kind of interspersing all these little clumps of nuclei. We think this is really, really important for controlling the state of the brain and shaping whether or not you can move into and out of different behavioral states. I'm not going to cover it an awful lot today, but I just want you to keep that in mind as we're thinking about these different circuits. Another area that is really important that we often forget about is the thalamus. So this is this little area, these little eggs that sit on top of the brainstem. You saw them in this picture before. Here, the thalamus sitting on like little blobs of ice cream on top of ice cream cone in your brainstem. And what they're doing is they're connecting to the entire cerebral cortical axis as well as to the cerebellum and many other subcortical structures. Parts of them are receiving sensory input from the world. Parts of them are controlled going down into the spinal cord. Other parts are receiving input from the tectum, which helps us shift our eyes around. And other parts are doing this much more sort of arousal-like function. And we'll talk about them some more, particularly a little bit today and particularly next week. This is the object that I told you a lot of people mistake for the brain. It's the cerebral cortex, this big lumpy external sort of structure that we see on the brain, split up into what we would call the different lobes. The frontal lobe involved in sort of planning, things that are protracted in time, and moving your body in a lot of in a motoric fashion. The parietal lobe controlling the integration between lots of different sensory systems. Let me use my pointer. The sort of somatosensory system, touch receptors in your body, the visual input that comes via the retina to the back of the brain in the occipital lobe, and the auditory input by your cochlea that comes into the superior temporal lobe. They all kind of find their way into this area, and you do a lot of multimodal integration. The other fancy word we used was affordance, which is the opportunities presented by the world, which you can then act on by planning with your frontal lobe what you ought to do in this context to achieve the goal you want with what's there for you to work with. The temporal lobe is a really interesting area that has auditory inputs, but also has this really interesting secondary processes where it takes the visual input that you receive and kind of categorizes it and starts to become more abstract as it heads up this sort of hierarchy. The other lobes we talked about, not to forget, the anterior or the cingulate cortex, which wraps around the cingulum bundle, this big white matter highway that runs between the two hemispheres. It has lots of different functions that you can think of as kind of supporting the cortical areas that live next to it. So in this subgenual cingulate, it's going to be supporting these more autofrontal functions like goal setting and inhibition. This sort of rostral anterior or dorsal anterior cingulate cortex is sort of supporting this sort of frontal planning and has a major role in salience detection. Motor cingulate for moving the body. Posterior cingulate ends up being involved in things like mnemonic processing and cognitive control. And this area here, hidden in the brain, if you pull apart with calipers, is the insula. This receives a lot of visceral information from the body. So things like the processes within your gut, and also has a big role in salience processing as well with the cingulate. We briefly covered the idea of these resting state networks, the ways that people parcellate up the cerebral cortex into sort of 7 little groups or 17 groups or whatever magic number of groups they like. And whether it might be a little bit better to think instead about these gradients of connectivity or organizing principles from these sort of more sensory systems, say in the visual cortex and the somatosensory cortex, sort of connecting in with this, what we would call heteromodal or more general associative cortex. And you could think about these in a really nice graphic as taking this really specific information from the world and then trying to integrate it in ever more abstract concepts to try to accomplish whatever goal state the brain is currently in at that time. This is the other, I just love showing this graph because I think it's such a cool thing. This is a really nice cladistic tree from Randy Buckner and Fenner-Creenan showing that these systems of sensory input and motor control are really highly conserved over evolutionary time. But what's changed over phylogeny is all these extra bits that are lying around, all these extra bits for connecting. If we were going to extrapolate that idea of the spinal cord, we've still got the channels running up and the channels running down. Now we've got a ton more of those horns, that gray matter between them that's allowing interconnection, and hence more sort of interesting and intricate types of plans and movements that we could achieve. And I had my stupid little LeBron James thing about the hippocampus, where if we tracked visual input from the periphery, where we've got these really detailed processes happening in V1 through the ventral temporal cortex, we end up finding our way into the retrosplenial cortex, the entorhinal cortex, and the hippocampus, the hippocampal formation. And so maybe one of the things that makes the hippocampus important for things like spatial navigation and memory is the fact that it has access to these channels that can come into it in many different forms. It can then sort of select between them or reinstantiate them as needed for their function. And the last thing we covered in what I'm now realizing was an incredibly dense lecture last time we talked, was this idea of these repetitive loops between the cortex and these subcortical structures here, the thalamus, the putamen and caudate of the basal ganglia, and the pallidum, which then recur across the frontal axis here, and the connections between the frontal lobe and the cerebellum via the pontine nuclei, which then go back through the thalamus to impact the cortex. So this idea of these recurrent loops falling underneath that hood of the gradient to shape the ongoing activity patterns in the brain. All right, so everything we talked about last time was really at this sort of gross level, this macroscopic level. We were trying to think about how this lobe was sitting in the brain. If I was looking at a brain scan, what would I see if I found a little blob in that space? But we also know, and we try to fool ourselves that it's not the case when we do neuroimaging, but we know this in our hearts, that this need not be the level of resolution that the brain ought to function at. In fact, it's probably a really, really, really poor approximation of where the actual functional circuitry of the brain lives. And so if we imagine going into, say, a single little millimeter of cortical tissue, we might imagine that there's many, many little cortical columns. This is the idea that you've got these nuclei within the cortex sort of organized in this sort of vertically oriented fashion, which then have this sort of horizontal distance between them. This is particularly evident in the sensory cortices, less so as we move up into the association cortices, and we'll start to talk about that. Each of these regions are getting fed by cortico-cortical connections, say from a different region, by thalamic inputs, and also by the neuromodulators of the ascending arousal system. We can imagine even zooming down further into, say, one of these different columns, and we could say things about different types of cells. Maybe there's a layer 5 pyramidal cell, which has got its dendrites sitting up in the supragranular layers. We talked about this in a complexity seminar that James ran. Or we've got these little CT-type cells that are sitting in layer 6 and only send their dendrites up into layer 4, which just so happens to be the major thalamic input to the cortex. And we can look at this level of resolution. You know, we need not stop there. We could go down to an individual axon or dendrite in a particular pyramidal cell, and we could look at whether or not there's different channels on certain parts of this, or whether there's dendritic integration happening in this portion of the dendritic field, or that proportion of the dendritic field. And you can go even further, right? You can go down to individual synapses and talk about the vesicles that are happening, that are releasing a neurotransmitter that goes across the synaptic cleft and causes some propagation of an active potential, or blocks a neurotransmitter so it acts for longer. So there's many, many different levels of explanation. Today, I'm going to talk a little bit about things on this level here. So thinking about how, at this sort of mesoscale level, how the circuits of this system are connected together, both within a circuit and also across the different circuits. Now, this is not going to be exhaustive, and if you are interested in this stuff, I'm happy to point you to references, or you can ask questions to get me to kind of sort of expose some of the complexity. This is going to be kind of like a bird's eye view of some of the circuitry, but hopefully it'll give you an appreciation for how the system, might start to sort of work together with the different constraints that are imposed by this architecture. All right. So before we get too much further, just a couple basics on terminology. So when you're reading about these different connections, you often see these terms afferent and efferent, and then you can think about systems as being either targeted or diffuse. I found a nice mnemonic for afferent and efferent. So afferent means that the activity is coming in, and efferent means that the activity or the connection is leaving. So we usually define this by saying there's a cell body with a set of axons that are receiving input, dendrites receiving input, and then axons that are projecting out to activate and create an action potential that passes through. It turns out this is actually a little bit arbitrary in a lot of cases, but we kind of gloss over that typically and designate one connection as the efferent input and then the afferent, the nerve coming into, say, some interneuron and then the efferent leaving. But so this is just a nice mnemonic. If you see this, so afferent arrives and efferent exits. So the afferent is coming in, the efferent's leaving. Just in case you sort of come across these things in the literature and you wonder what on earth people are talking about. Another really important distinction when we're thinking about how these systems are organized and wired is whether or not they're utilizing different classes of neurotransmitters. So neurotransmitters are these little chemicals that are stored in vesicles in synapses and get released into, say, a synaptic cleft or into a cell body or into, say, the spine of an axon that can actually change the way that the system is working, the way the cell's working. Now, this isn't not an exhaustive list, but the major classes that I like to think about when I'm trying to work out how the system might be organized is these sort of more primary motivators, these ones that can actually cause the system to act in a particular way. So glutamate is the main excitatory neurotransmitter in this class. And so if I put glutamate into a synapse, it's going to hit a receptor, a glutamate receptor, and likely cause the cell to fire. There's some caveats to that, but that's the basic idea. Whereas if I put GABA in, then GABA, and you'll notice as well that GABA and glutamate are extremely similar chemically. The big difference is sort of on this end with this little sort of oxygen groups on the carbon. What that's going to do is typically activate chlorine cells, which are going to, chloride channels, which are going to allow chloride in, which is going to lower the potential and cause the cell to not fire. And so we really think about glutamate as promoting firing and GABA as inhibiting firing on this sort of like primary processing level. But there's this other really interesting class of chemicals. Again, I said this is non-exhaustive, but these are sort of main categories people typically think about, which we would call neuromodulatory chemicals. And so these guys aren't going to come in, forget about acetylcholine and serotonin just for now, because they've got a big asterisk on them. But the big idea is that what they're going to do is they're going to hit receptors that are, instead of opening a channel, are actually going to change the transcription within the cell. What they're going to do is they're going to come in and they're going to cause a cell to upregulate genes, which will then open up more calcium packets and let the cell burst a little bit more. Or they'll say, actually, we've been firing a lot. We could use a bit more receptor help. So upregulate those receptors, give me more of those receptors on the other side of the synaptic cleft. So they're going to have these changes that are going to happen at a slightly different time scale than the ones that happen with glutamate and GABA. These are going to happen really quick, and we call these ionotropic. And these are going to happen quite slowly, and we call them metabotropic. Now, the caveat here, there's always a caveat with biology. Why does it have to be so damn complex, right? Is that acetylcholine and serotonin both have receptors that are ionotropic as well. So these use a nicotinic receptor, which acts really quickly, and serotonin 5-HT3 receptors are ionotropic as well. So the acetylcholine and serotonin are super interesting because they can have effects on both really quick and really long time scales, whereas noradrenaline and dopamine are much on this sort of more slow time scale. Another important point to note is that there's lots of different classes of receptors. Here I'm showing the difference between an AMPA receptor and an NMDA receptor. There's many different classes. Each of the different neuromodulators has got its own subfamily. But the idea here is that AMPA receptors and NMDA receptors are both using glutamate to cause an influx of chemicals that then cause the cell to change its potential and cause it to do an action potential. But the funny thing about them is that NMDA receptors have got this little ion sitting in the channel, blocking it from acting, unless the polarity in the cell changes a little bit. So unless a few of these AMPA receptors have come in and nudged it on a little, which then causes that calcium channel to flick out. There's a couple other cofactors that are important as well. But what that then means is that lets in a huge amount of the neurotransmitters that cause a burst in the cell. So you can see that there's already these levels of nonlinearity baked into the circuitry from how the system is connected. All right, so any questions at this point? All right, so the cerebral cortex. So before we launch into the cerebral cortex, I thought it would be important to note that I made a mistake last time that James Henderson quite nicely told me. I said that if you flattened out the brain, the cortex, you'd get something the size of a tennis court, but that's complete garbage. That's your lung. It's actually about the size of a rug. So it's about 2.5 meters squared in each hemisphere. So that's important. I just want to make sure that, you know, I wasn't pulling it out of my lungs. Okay, so if we look at the cerebral cortex, one of the ways that we used to carve it up back in the old days, Corbini and Brodmann was one of the most popular approaches, but the Votes, the husband and wife group, along with a number of other really precise neuroanatomists, what they would do is they would take the brain and they would look at little slices under a microscope, and they would try to carve it up into these different regions when what we would call the cytoarchitecture changed. So here, the architecture of the cells, they would say, okay, if I'm sitting here in this area, we'll call it area 46, everything looks pretty similar, and then I track across a little bit, and all of a sudden, things have changed. Now there's more granular tissue, there's more cells in this layer than that one. And if I move again, then I'm going to find a different sort of cytoarchitecture. And so when you look through the literature, you might see things like these Brodmann areas. This was a really, really popular way to describe results, particularly in early neuroimaging, because it was the sort of reference point that people could use. Oh, you know, I care about language. I know that Brodmann area 45 is really important. So, and, you know, let's say parietal lobe 39 and 40. So I'm going to tell you whether or not I'm in 39 or 40, and then we'll somehow have learned something about the system. It turns out that it's actually a lot more complicated than that, unfortunately. But this basic principle is really important, that the cytoarchitecture across the brain, is particularly if you look along this rostrocaudal axis from, say, the primary sensory regions up into the association or heteromotor cortices, tends to differ an awful lot. So what do we mean by cytoarchitecture? So it turns out that if you look at the cortex under a microscope, if you pulled it out of the brain in an autopsy, it's actually a really thin sheet of gray matter. It's about the size of 5 or 6 playing cards, and you've got all this white matter underneath it. And if you looked at those playing cards under a microscope and you stain it in particular ways, which is another really important point, you can typically break it up into what people kind of loosely agree is about 6 layers. Now, this is like there are 7, resting state networks in the brain. There's no such thing as 6 layers. It just happens to be a convenient way to parse up the system. In fact, if you go and look at other parts of the cortex, some people say, actually, there's 17 here, and there's 14 in this area, and there's only 3 over here. So this is just a sort of, as I said, a bird's eye view to kind of give you an idea about how people parse these things out. So I said there's basically these 6 different layers. That the reason that we can parse them up is that they contain different concentrations of cell types. And so, what I'm showing here is sort of this basic kind of idea of like these big pyramidal cells in the cortex, which have their cell body sitting in a layer. And they have what we call apical dendrites, usually spreading out locally, horizontally. And they have these, they're basal dendrites, I apologize. And then they have these big, long channels, leading up to their apical dendrites, which sit in the apex, up near the cortical surface. So this would be like the skull heading down to the thalamus and brainstem down here. And they're receiving inputs on these different dendrites from different axons, either locally or from distant regions of the cortex, and then projecting out either to other parts of the cortex, to the brainstem, spinal cord, basal ganglia, colliculus, or to the thalamus. And so there's these particular organizing principles in terms of how the layers are organized. Now, another important thing to note is that instead of describing things at these 6 layers, there's actually another sort of nice way to do it. Layer 4 is typically called the granular layer. And the reason for that is that when you look at it under the microscope, it typically stains really, really thickly and densely. On this next slide, you can see this quite nicely just over here. So you can see this really dense, what people would call granular layers, because it looks, there's like the real granularity to it. That's the area, along with some of the lower parts of layer 3, that receive lots of input from the thalamus. And this will come up when we start talking about the thalamus. Below that, so we call that the granular layer. And then below that, we can be quite convenient. We can call this the infragranular layer, the layer that's below the granular layer. That's going to have these big pyramidal cells, typically layer 5 pyramidal cells. And we describe the region by the layer that its cell body is sitting in. And these layer 5 pyramidal cells in the motor cortex are the ones that project down to your spinal cord and control whether or not you wiggle your feet and your hands around and move your neck. But in other parts of the brain, it project into things like the basal ganglia, the pontine nuclei, the superior colliculus. And then these areas are in layer 6, which is also a supragranular layer, but it's, infragranular layer, but it's lower than the layer 5 cells. These are what we would call the CT, or the corticothalamic type. And these are the ones that are projecting down to the thalamus and receiving input back from the thalamus, and give you this nice little resonance loop with a region of the thalamus, these nuclei called the core nuclei. Now, if you were going to call this infragranular, then it makes sense to call this one supragranular. So these are the layers that are above the granular layer, and they typically contain these other population of what we would call IT type, or intratelencephalic type pyramidal neurons. And what these neurons are typically doing is projecting both locally, horizontally, but also forward up what we would call the hierarchy in the cortex. And so they're really doing this projection to other cortical areas. So in a very heuristic, and just as a final point, layer 1 is kind of like a funny layer because it mainly is mainly comprised of white matter. So it's got all these connections from different regions along with a couple little inner neurons. But so at a very heuristic level, we can think about layer 4 as receiving input, layer 5 and 6 projecting output out, and layer 2 and 3 is sort of integrating across the top. That's sort of a very high level. So another thing that I mentioned before that's really important is that if I looked, say, at the cytoarchitecture in the sensory cortex, so this is the primary sensory strip and the primary motor cortex, they're quite different. You notice that in the sensory cortex, you have this really dense granular layer, this big fat layer 3 cells and small layer 2 cells, but really proportionally those regions are much smaller than they are in the primary motor cortex. And you notice in primary motor cortex, they've got this huge, what we call a BET cell, which are these layer 5 pyramidal cells projecting out to the spinal cord. And so there tend to be these systematic differences across the neuraxis. People typically say that they go from what we would call a granular cortex to an agranular cortex. So this might be what we might find in the anterior insula or part of the cingulate cortex, whereas over here is going to be in the more sensory region. So you can see this really thick granular layer receiving the thalamic input, and over here you've got these sort of cells that can activate and go off and an action, and they have these regions that are integrating, but really aren't receiving a huge amount of thalamic input. All right, so this is just like a little diagram of 3 of the cells I've mentioned so far. So the corticothalamic type, the pyramidal type, and the intratelecomphalic type. I wanted to make the point again that this is a massive oversimplification, and it reminds me of this really interesting idea that in science you have either lumpers or splitters, and this is massive lumping. Like this is hardcore lumping, and if I met a splitter, they'd like come in and punch me in the face for saying this stuff to you. But I wanted to give you this kind of like high level understanding of how the system might be comprised. So you've got these different cell types, right? And so one of the really cool things is that if you go and look, and you have to use sort of studies that go across different species to make these inferences, so they'll always come with a grain of salt, as well as things that causally impact the circuits, such as optogenetics, to try to unpack some of these different these circuit functions. But one of the really cool things is that these circuits typically do have a sort of pattern of rules that define how these different neural types are connected. And so I mentioned before that we might have this thalamic input. Let's say it's coming from the retina via the LGN, and it's going to hit this granular layer of cortex. Now, there's another type of cell here called a stellate cell, but for convenience, I've removed them. But the idea is that they're going to hit a lot of these apical dendrites of this corticothalamic cell, which will then, if enough of them get activated, it'll cause the cell to come down and do like a little resonance loop. One of the other things that happens, so there's that little thalamus happening. Another thing that happens is the stellate cells pass that activity on. They're connected into these supragranular regions, layer 2 and 3 pyramidal cells, which then feed the activity forward, both locally within its column to the layer 5 PT type, but also they also feed forward to the granular layer of the next region up the hierarchy. So the idea would be that the thalamus is pumping in a bunch of activity to this region, but so is this layer 2, 3 IT type cell. So they're going to be integrating that input and deciding whether or not to resonate. The PT type is going to do its little, send its little efference copy off to the subcortex and brainstem, cerebellum. But it also does this thing where it feeds back from a more agranular to a more granular region. And it tends to feed back and make projections back to the supragranular layers. Those layers I said were mainly white matter. It's where all of the dendrites of the layer 5 pyramidal cell and the layer 2, 3 cell are sitting in a previous layer of cortex. So if it activates, it's saying, I'm going to go find out what the cortex, the subcortex, and the brainstem said I should have done, and I'm going to go back and tell everyone what I just did. So they're kind of like a bit of an a-hole of a cell, right? They're not really doing all that much work. They're getting their friends to do all the work for them and then bragging about how much work they did. Everyone knows these cells. All right, so one of the really cool things is that that gradient that I showed you that you can estimate from resting state fMRI actually does look like it gets recapitulated in a lot of the cytoarchitecture. So if I look at a relatively granular layer, right, I see this thick granule cells and really small cells in the supragranular layers, quite sparse layer 5 pyramidal cells and lots of corticothalamic cells. But if I move forward in the hierarchy, recall that we were moving from sensory regions to these more heteromodal regions, what I start to see is these really big, dense layer 2, 3s that have big cells, and these cells actually have lots and lots more connections individually than these cells do, and relatively sparse granular layer, and still the same corticothalamic cells, but way more of these layer 5 PT type cells. Some really cool stuff's coming out of neuroimaging. This is a really beautiful paper in PNAS just recently by Branislav Misic's group, where they were actually able to show that this same gradient is associated with what they called structure function tethering. So the structure of the white matter connectome in these blue regions, which typically are those ones low on that gradient and are more granular, are much more reflective of the underlying structural connectivity. So if I told you what they were connected to structurally, you'd be able to tell me something about their activity pattern. As you moved away from those regions into more heteromodal cortices, you start to lose that explanatory power with the structural connectome. Ben Fulcher, who's not here today, has shown some really cool stuff where that same gradient is consistent across humans and mice. It typically goes along with this thing we call the myelin gradient, or an indirect estimate of the myelin gradient, which is how much white matter there is in the different parts of the cortex, which I think is super cool. And then a bit of shameless self-promotion. I've been working with Joe Lizier, Oliver Cliff's been working on some work where we show that these different regions, these sort of supragranular, granular, and infragranular regions, typically have different information processing signatures, such that the supragranular layers in rest are kind of receiving a lot more input from the granular layers, as if you're sort of sitting there passively soaking in the sensory input. And it looks like, I don't have the slide here to show it, but it looks like the whole pattern flips over when you start to do a task. So all of a sudden, the supragranular regions are the ones that are actually pushing out. They're having the influence over the sensory regions. So it suggests that this gradient idea might be really informative for the functioning of the brain. We shouldn't parcelate it up too early, but instead ask whole brain questions and see whether or not they are reflective of the underlying cytoarchitecture. Another really important point, I don't have exhaustive slides on this, but I just wanted to kind of put the idea in people's heads. This is a beautiful slide for the fact that this figure, for the fact that it is completely overwhelming. This is supposed to show you a pyramidal cell and all of the different interneurons. These are called smooth cells. These are the little supportive, typically GABAergic cells. So the pyramidal cells are predominantly glutamatergic. They're going to go off and excite. Whereas all these little interneurons are the ones that are going to come in and basically keep it under wraps. They're going to tell it to be quiet at certain points in time. And there's just so many of these different things. And they're all weird and wonderful. Some of them are like these little lurky little sort of things. It almost looks like something you'd see at the beach, right? When you walk down and look at different types of seaweed. Some of them are long and thin, and others are short and fat. And they've all got different calcium binding proteins and different levels of activity, different parts of the cortex that they want to live in. So these are just adding like another layer of complexity on top of the circuit that we've already oversimplified. So it's just another point to remember that we are definitely in Clumpville. If you are interested in getting a kind of high-level intuition about what these might be doing, one really interesting thing is that there's only really 3 major classes of these different interneurons. If you stain them for different calcium-binding proteins and look at their genetics, you can very easily split them into these 3 major groups. A parvalbumin-positive group, the basket cells are a good example of these. A somatostatin-positive group, there's the neurogliaform, typically here, and these VIP, which are also known as a 5-HT3. They're really exquisitely sensitive to serotonin 3, that ionotropic receptor I mentioned. That, there's an example of one of those guys there. And so the idea is that people are starting to work on this notion of this sort of relatively low-dimensional structure where you've got a parvalbumin-positive cell. Other people have shown this is really important for creating gamma synchrony. What it does is a pyramidal cell activates the fast-spiking cell, this parvalbumin cell, which then tells the pyramidal cell to shut up. But then the pyramidal cell overcomes that inhibition and spikes again. And so you get this constant back and forth of excitation inhibition at these sort of gamma frequencies that might underlie some of the signals we see in things like electrophysiology. So these parvalbumin cells are then inhibited along with the excitatory cells by somatostatin cells, which are in turn inhibited by these vasoactive intestinal peptide cells. And so you've got these really interesting circuits where you can kind of see, here at a very fuzzy resolution, that you could start to kind of put the brakes on and shape the activity that can emerge from these circuits at a higher level of granularity than you'd have if it was just feed-forward circuits running on their own the whole time. For people that are interested, this is an absolutely spectacularly beautiful paper by Carol Svoboda's group. what they did was they got Cree lines for the 3 different populations of inner neurons in barrel cortex. And barrel cortex is the area of the brain in a rodent that is sensitive to the movement of the whiskers, right? So they've got little whiskers flicking around, and the barrel cell pyramidal cells, people have shown this little purple guy, are sensitive to the relative phase difference in these whisker movements. And so the idea is that the mouse whisks its whisker across something in the world, and then relative to how much it moved, how different the phase is, it can determine what was out there in the world. It's sort of its way of detecting what's in its environment. And so what they did was they basically were able to turn on and off and record from all these different populations of inner neurons within this barrel cortex. And it's way too complicated of a paper to unpack here, but the basic idea was that the parvalbumin cells, these fast spiking cells, were really important for sensory recognition. And so were the somatostatin cells. That was a bit more contextual recognition. But anytime the animal did something voluntarily, moved their whisker, these vasoactive intestinal peptides are incredibly important. So it might be that at this level of granularity, we might actually start to be able to unpack more passive versus more active processing. And as I said, there's a lot in this paper, so if you're interested, go have a check it out. Just to sort of ram the point home, that I think is super fascinating, these different calcium binding proteins, which I've said before represent different inner neuron groups, are actually differentially expressed across the brain as well. And if you squint, you can almost imagine here that we're low on this sort of low dimensional gradient here. We've got visual, occipital, and more sort of sensory regions. And as we move up the hierarchy here, it's not exactly the same as the map that we showed you before, but you can see that in these sort of more agranular and limbic regions, as well as in this cingulate cortex, you see a relative expression of these different other receptor types. And so it might be that gradients in the expression of these cells imbue more of the kind of volitional goal-directed control in these more anterior circuits and more of this sort of passive sensory reception in the posterior circuits. All right. Questions about the cortex? Yeah.
It looks like there is some basal neural activity in the cortex, resting state and that sort of thing. Where that synchronicity is coming from? How different bit of the cortex can work together in a fairly synchronized way.
Yeah. Okay. So like any good politician, I'm going to answer the question I wished you asked rather than the one you did. So when we measure activity in the brain, we are measuring very particular events. We're measuring summations. Typically, we say local field potentials, which is where we get electrophysiology like EEG, MEG, but the bold signal as well. We're measuring the summation of multiple different synaptic events that have occurred at the same time that also happen to be in the same orientation that then comes up to, say, the surface of the brain and then amasses enough that we can measure the potential with an incredibly insensitive little sensor on the surface. So the pattern that we see on, let's say, an EEG is not reflective of the circuit. It's reflective of a particular part of the circuit in a particular mode of firing. It's probably going to be something like a whole bunch of those big layer 5 pyramidal cells firing all together en masse at some frequency that then brings in, lots of calcium changes and brings in lots of blood flow. So we see bold as well. That's going to probably explain something like what we measure. So I think, it's very hard to, in other words, it's very hard to answer your question because what we have is an indirect measurement of a really complex system in action. So it's probably going to be something like masked activity, but if we have other more sensitive measures, if you've seen things like the neuropixels probes or voltage sensitive dyes that can pick up these layer specific differences, then it's going to be really interesting to see how the circuit starts to work together when you can measure all of it rather than just the part that you get from this sort of summed activity. We can talk later if that isn't satisfying. Any other questions? All right. So the thalamus. So I mentioned before that I'm a big fan of the thalamus, and I think we all ought to be because it's probably one of the most beautiful areas of the brain. But it has been long and oft forgotten, partly due to a similar reason to what I've described just then in the answer, which is it's really hard to measure it. Right? We know that if someone gets a stroke in their thalamus, then they're going to be in big trouble. We know that if we stimulate parts of the thalamus, then we get really big, obvious behavioral effects. So we've known for a long time it's very important. It's quite metabolically active. We know that when people wake up from sleep, so when the arousal system dumps a whole bunch of neurochemicals into the thalamus. It moves into a different firing mode by opening certain calcium channels that weren't before, and you get this completely different dynamics. It moves from a burst firing mode into a tonic firing mode, and that's really important for the things like arousal and attention. Masayo Stereard has done absolutely beautiful work, 30 odd years ago. That's typically kind of just passed by most of the people in my world that think about, whole brain imaging. But it's incredibly important. And one of the reasons that it's going to be really important is due to its connectivity. So we talked last time about these sort of different nuclei, subnuclei within the thalamus. Here we're showing things like the pulvinar, which is connected to the parietal lobe, the lateral nucleus, which is connected to the occipital cortex. But really every part of the cortical mantle is supported and supporting a thalamic nucleus. So this is just a nice figure from Lawrence Ward's paper that shows that, you can think about frontal cortex and moving back to the motor cortices and then back to the temporal lobe and the parietal lobe into the occipital lobe. And every one of these regions is sending projections down and receiving projections back. Another really important bit that I'll talk about in a second is this reticular nucleus. I found out recently at a neuroanatomy class that we shouldn't call it the reticular nucleus of the thalamus because it's actually not part of the thalamus from an embryological perspective, but it's definitely part of what we call the diencephalon, which is that one of those little bumps that I showed you on that the neural tube as it starts to form. So the basic circuit in the thalamus has been known for a really long time, and a lot of the work here has been done, and I think it's important to realize that it's been done in visual cortex. And so before I told you that visual cortex is a sensory cortex, it's a really granular cortex. It's got a big, thick granular layer. It's got really small pyramidal cell layer, lots of CT-type pyramidal cells. It's a really feed-forward system. And so that means that you're going to get a lot of the population of cells that have this CT type, right? This is our little corticothalamic type cell with its cell body layer 6 and its projections, its apical dendrites up in layer 4, the main receptive window from the thalamic cell. And what it's going to do is it's going to project its axon down. It's going to hit typically quite distally on a thalamic relay cell. People call this like a thalamic relay nucleus, even though that's probably imbuing a function on it that's maybe not 100% accurate. And then that thalamic nucleus will then excite, this is an excitatory projection, will then project back up into the cortex into that layer 4, where it'll hit stellate cells, parvalbumin cells, as well as that layer 6 cell. On its way, it projects a little excitatory connection into this little red guy here, which is the reticular nucleus. And reticular, I believe, means shield in some form of ancient language. And the idea is that the reticular nucleus is sort of surrounding the thalamus in this little shield of inhibitory connections. So rather than the excitatory connections of the layer 6 and the layer 4 cell, this is a GABAergic connection. And the idea is that anytime you activate, you remember before I showed you that little loop of pyramidal to parvalbumin and back and forth, you're going to get a similar kind of thing here. If this little guy, this little layer 6 cell starts to fire repeatedly, as it's firing and trying to activate this little guy, the core thalamic nuclei, it's also going to make it become inhibited. And so there's some really interesting optogenetic works that's shown that will actually cause the cell to fire in something between a kind of 10 to 40 hertz range. Depends on the kind of state of the cortex when you stimulate it. The thalamus is also receiving a bunch of ascending afferents, and this is really important. This might be something like an axon from the retina, and you'll notice that the projection here is actually much closer to the cell body. This also could be something like an axon from the cerebellum or from the superior colliculus that's going to drive the cell. And so it doesn't actually need to be activated by this layer 6 cell type. It could be activated by a subcortical route, which then causes it to fire. And if the milieu is correct, it might then drive this back down and cause a little resonance loop for the time that it's driving this ascending activity. All right, so when I told you about this a few minutes ago, I kind of made a big point about the fact that we were sitting in sensory cortex here. So why is that important? Well, the reason it's important is that the thalamus is not homogeneous. The thalamus has these at least two, and very, very likely more. This is like, I should put like a little sign up that says clumping alert every time I oversimplify. But this little circuit we've just talked about would fall into this green category, right? We've got a cell that's, a nucleus projecting into layer 4 of the cortex, and then maybe it receives a projection back. And so Ed Jones called this the core type. They're really densely stained cells. They're typically clumped together, but they're interspersed by a completely different population of cells, which Ed Jones called the matrix type. Other people have called these the non-specific thalamic nuclei. Others still in the subpopulation of these are called the intralaminar nuclei because they sit between the lamina of these, in the thalamus, you can sort of see these like little lamina that run through it, and they sit kind of embedded within that often. But these matrix cells, instead of doing that little targeted projection up into the granular layer and getting the feedback from the layer 6 cells, what they actually do is they project up diffusely often to the supragranular layer. Right? That was the area I said, the supragranular layer is one which has got all this white matter connections and all the feedback coming from higher regions back down to lower regions, and also sending projections back up. And so it's doing this really integrative role in the cortex. And what these thalamic cells are doing are almost like raising the temperature of those cells and causing them to be a little bit more active, but in a much different mode than the activity you would get if a core thalamic cell started to activate. So you can imagine here, I show you a face and your LGN goes, oh, I saw a face. Hey, I saw a face. Oh, look, there's faces here. And we start to resonate away versus, uh-oh, something's looming. I don't quite know what it is. Quickly get the system on board. We need to work out whether or not this is a problem. Right? So they're very different firing modes. Just to sort of show you some of the nuance, here I've got these different thalamic types. This here would be our little core type, the one that was in light blue before. This is a really nice paper by Klasker colleagues. And it shows that this core type is projecting up into layer 4 with a little offshoot into layer 6. The matrix types can be projecting up and really doing this really diffuse, often completely global, but more frequently a more sort of constrained but still relatively diffuse connection. There's also a few cells, particularly in the medial dorsal nucleus and the pulvinar, that actually have a relatively targeted input to the supragranular layers along with the layer 2-3 cells. And then there's this other little category. I think they've oversimplified the IL here. The intralaminar nuclei do actually project up to the supragranular layers. But they do something really fascinating. They send this massive, like 100 to 1 compared to their connection to the cortex, input to the striatum, the basal ganglia, and the amygdala. Yes? How long's a piece of string? Some of them are relatively local within their own column. Some of them go, honestly, over the whole lobe. And so there are some estimates that in rats, I don't know if this actually extends to humans, that certain of these multi-areal matrix types actually cover the entire cerebral cortex with a single axon. So they're doing something a lot more like the arousal system than this core type, which is going to be propagating this sort of more specific signal through. Yeah.
Do they sort of fire at different time scales? Like the matrix ones sort of maybe have a slower time scale that kind of set up the vibe and prime the cortex, and the core ones sort of have maybe what we might call a bit kind of basic, short of the. Yeah,
so it's a really good question. And I don't think that the appropriate work has been done to really break this down. Some really interesting work has been done in the lateral geniculate nucleus, which is, the vast majority, like 90% of work in thalamus has been done on the visual system. And there, the matrix cells are typically in a class called the coniocellular cells so if you look at the LGN, oh yes, I'm gonna use the whiteboard.
Yes.
Sucked in recording at home. Right, so the LGN has got these little boomerang-looking things, which also look like a bend, which in Latin is articulate, thank you, Brandon, which contain all of the core cells. And they're going to be in two different classes. There's a parvocellular and a magnocellular cell. The parvocellular cells are typically selected for things like color, and the magnocellular to track things like movement. And then interspersed in the background are these coniocellular cells. And the K cells typically have a much more diffuse input. They get visual input from the colliculus, but also from the retina, and they project much more diffusely. Now, Brandon, do we know if their timescales are different? Did you guys look at that in your stuff? Yeah, K is slower. So K is slower. So the matrix cells are slower. Makes sense. You want to have selective input. I saw movement now rather than in 5 milliseconds versus something bad is going on right now, or this is a good place to be. Brandon, if you're curious, Brandon's done some stuff that he could pass on to you with Paul. Okay, so one way we can make purchase on this in the world of zoomed out world of fMRI is we can try to get an estimate of the relative loading of these different populations in the thalamus in a human that's lying in a scan. And so this is typically hard to do. You can't go in and take a punch biopsy of the thalamus and just pull a bit out and say, oh, there's a bit of matrix there or a bit of core. One thing we can do is we can actually use a really interesting indirect measure, which is those same calcium binding proteins I told you before delineate the interneurons of the cortex. So in this case, the core cells are heavily stained by a chemical called parvalbumin, and the matrix cells are heavily stained by a chemical called calbindin. We don't think they have a functional role. They're both just signaling the fact that calcium's around. So there's calcium everywhere, but it just so happens that different genes probably turn this one on in this group and this one on in that group. And so what we were able to do was we were able to take from the Allen Brain Atlas the relative staining of the mRNA that supports calbindin and the relative staining of the mRNA that supports parvalbumin for each voxel within a 7 Tesla fMRI study. So that has a really nice spatial resolution. And we could map the little time series of these nuclei, these little voxels over time. And we could ask, are there any brain regions back out in the sort of cortical level that are tracking more with these matrix type cells or more with these parvalbumin type cells? And yes, there are. And if you have been listening so far, you'll notice that this is looking at one end of that gradient, and this is looking like the other end. So it looks like the cells that are in the thalamus that are going to be projecting the sort of sensory-specific information back up to the cortex are really heavily enriched in the areas that have really thick granular layers and are processing sensory information. In contrast, the areas that are rich in calbindin staining and have lots of matrix cells are going to be much more densely innervating areas that have rich supragranular layers, which are the ones that are associated cortex in the frontal cortex, the inferior temporal lobe, and the lateral parietal lobe. But if that doesn't convince you, that's the correlation with the loading of the primary gradient from Margulis. I mean, it's quite astounding just how well it recreates the pattern that you see when you just cluster vertex-wise time series in the resting state. You'll also notice that's not going to be the whole story. These are not linear relationships. I put a linear line on just to show you, but I mean, that's like quite a fascinating little pattern that's going to be worth thinking a little bit about. Why is it that it's loading heavily onto some of these regions and not others? And then, the other interesting thing to think about is that some of these primary gradients are interesting, but then a lot of functional differences start to happen as you start to get down to these lower gradients. So it'll be worth thinking about how these things relate to one another. But I wanted to close today's lecture by making a hat tip to somebody who saw this coming probably going on 20 years ago. Marcel Messelin has this beautiful paper in Brain in 1998 called From Sensation to Cognition. And what he basically argued was that we should, instead of thinking about these different subunits of the brain and little specific functions, we should instead be thinking about this gradient between the way that we can experience the world and whatever it is we're trying to fix in our world right now. And so this is a little diagram of Maslow's hierarchy. And the idea here is that at the bottom, you've got things like physiological needs, like right now I really need a glass of water, or maybe one of you has to use the bathroom or something. And as you start to put those to bed, and you can actually become focused on different levels of abstraction to the point where at the top you're a self-fulfilled person, a self-actualized person, fully achieving your vision for yourself in your weird world. And the idea is that what the insight that Messelin had was that instead of thinking about these little individual bits, we should think about these regions in between this, what he calls the heteromodal cortices, as integrating between the options in the world and the things that we're trying to achieve. And I think he was totally spot on. And he called it 20 years ago before anyone was doing proper neuroimaging. So if you ever see him at a conference, buy him a beer, because that's an awesome prediction. So with that, I want to leave us there. And then I'm going to skip ahead to the very last slide to say I had way too many slides. Next week, I'm going to talk about how some of this all fits together. And where that's going to start is in one of the little things I teased before, which is that the thalamus is receiving input from not just the cortex. It's receiving input from the basal ganglia, from the cerebellum, from the retina, from the colliculus. And I think that understanding that and putting that under the microscope is going to give us a real good handle on how it's going to shape cortical dynamics, which will then come back and shape the thalamus, and shape the systems that it's getting activity from. And so I'm going to try my best to put it all into a little story next week that we can then discuss. So yeah, thank you. So, who has questions? Yes.
How do the thalamus learn that it's a relay, it's a filter, and it's a switch? Does thalamus really make any processing at all? And how you can differentiate what thalamus does to what cortex does?
Are you a corticocentrist? So yes, I don't necessarily follow what you mean by process. I think that if one thing that we've learned from deep learning is that you can get an awful lot of purchase out of having a bunch of blind little units connected in interesting ways that then get fit to opportunities, let's say shapes of the data or things that you want to do. And so I think I'm probably in a different category than most. I think that the processing is a distributed process rather than a localized process. I don't think that a little message gets past the thalamus and it goes, ooh, I'm going to turn it this way and then send it back to someone else. I think the thalamus plays a crucial role in shaping the way that the rest of the system can work, but only in the context of the whole system can the function arise. So it's just a different way of thinking about it, but I don't know how to answer your question based on that frame of reference. But hopefully after next week, you'll see about how I think that it could do it. But it's not just a relay.
But in the slide that you showed from your guys, you and Brandon's work with the projections from the different sort of cell types in the thalamus and the resting state, it looked quite binary, like they're really projecting specific, different types of, I guess, regions. To what extent is that, thresholding things? Actually, they are really actually, mixed in where they're projecting the cortex, which would kind of make sense. But then if you really try to kind of look where the heaviest projections are, you see that separation.
100%. So what we did was we correlated the each voxel in the thalamus with every region in the cortex. We parcellated, and we got a relative score for how much it was like the parvalbumin weighting, relative score for how much it was like the carbinum weighting, and then we flipped them over and looked at the difference. But they're all relatively positively correlated. The thalamus is doing whatever the cortex is doing most of the time, but it's more, if you're moving to the more matrix-rich areas, it's doing something more like the associative regions and vice versa. But yeah, it's not, the binarization was purely just a statistical artifact. Cool. All right, so if people have questions, so far I've just been sort of guessing what people want to hear and mainly just selfishly talking about what I like. So if there are things that you want to know about, please email me or get in touch with Archive and just say, hey, how come you're not talking about hippocampus? And I'll be like, I don't know about hippocampus. And I'll get someone who knows about it to talk about it. But if there are particular areas of interest or there's something you're like, oh, how does that work? Next week, I'll probably try and save a bit of time at the end for discussion and kind of that little window where you can be like, hey, what's that? And if you tell me ahead of time, I can find slides and GIFs and stuff. Cool. All right, thank you. What do I do?
We'll just stop the recording.
Stop the recording.
So if you go to more, stop recording. Do you want to? Yeah.